As you know, this is our last lecture for 2016 of TRACO, and I'd like to acknowledge Jonathan Ruiz for providing the financial support for the course, especially the video casts, and Anthony Thomas here in Building 50, as well as Chris Graham in Frederick for setting everything up for the video casts. And our speaker today, our first speaker is Kurt Harris. He got his MD degree from the University of Kansas, and he did a residency at UCLA. And he's been at NCI, he says, for over 40 years. So he's chief of the Laboratory of Human Carcinogenesis. His title, Lung Cancer Precision Medicine Strategy. Take it away, Kurt.
Thanks, Terry. Well, it's good to be here again. And I want to thank Terry for managing this course for quite a number of years, and it's been quite an important one. So I think we all appreciate it. So let's proceed. All right, precision medicine and lung cancer. I'm sure everyone in this room and on the video cast understand that the main cause of lung cancer is cigarette smoking. And in fact, there's been somewhat of a decline in the incidence of lung cancer in males and maybe in females. But over the last 50 years, from the Surgeon General report, which I and a number of people contributed to, there's been more than 20,000 premature deaths associated with smoking. And that includes cardiovascular disease, pulmonary disease, and of course, cancer. So in terms of cancer prevention, anti-smoking efforts are probably the most effective in terms of decreasing the number of deaths of cancer. Well, while smoking has gone down in the United States, that's not true in other parts of the world, here in Chile and especially in Asia and the Soviet Union and part of Europe, in which smoking is still very high, in some cases around 50 to 60% of the populace. So lung cancer is going to be around for quite some time. It's obviously the most lethal type of cancer worldwide, and that's followed by liver and colorectal and breast and some of the others. And one of the reasons I've studied lung cancer for quite a number of years is the fact that it is the most lethal and the most challenging in terms of therapy. But there's some hope, and that includes identifying people who are at early stage lung cancer. And I'll be talking about low-dose CT and the use of that screening strategy. And there, people who are at risk are obviously smokers. And one of the objectives is to identify people in this category, and there's about 10 million of them in the United States, who need priority screening by low-dose CT. And I'll tell you about why that is the case and something about this kind of test. And so there's a need for screening biomarkers of lung cancer risk. There's also, once the tumor has been identified, is it cancer or is it not cancer? And that means that one needs diagnostic markers. And then once it's surgically removed, some of these people will have recurrence, and I'll talk about that and how can you identify those people who have a high probability of recurrence versus those who will not. Those are prognostic biomarkers. So the new initiative in precision medicine goes back just a few years now. It was, I think, really stimulated by a review by the Institute of Medicine which was entitled Precision Medicine, which I'll get to in a moment, was announced by the President Obama in his State of Union message, and then this is an editorial written by Francis Collins, director of the NIH, and Harold Varmus, who was at that point director of the NCI. So the traditional kind of medicine, the kind of medicine that I learned and actually practiced early on, was that one does an interview of the patient, and I guess you would call him or her a client today, and get a medical history, and then do a physical examination, and then do some standard laboratory tests and develop a differential diagnosis, and then a treatment or exploratory procedure. What's precision medicine? It's multi-layered, it's individual-centric, and it's interconnected. And it uses information commons with these various omics that is built upon the base of clinical information and epidemiological information. And I'll go through this in more detail. So in this report that came out, the National Research Council in 2011, the information commons includes the expososome, which I'll talk about, the genome, the metabolome, epigenome, transcriptome, microbiome, and then the clinical information. And this is utilized to develop the knowledge network and a new molecular taxonomic classification of individual patients. And the objective is to improve clinical medicine, diagnosis, healthcare, treatment, and with the feedback going to the information commons of observational clinical studies. But it's also informed by medical research, and the feedback loop is informed mechanistic studies. And then something that was not included in the original strategy was cancer prevention and to guide preventive research. So let's first start with the expososome. It was coined by the now director of the International Agency for Research on Cancer, Chris Wild. And when you think of exposure, normally you would think of just the external environment, tobacco smoke, infections, diet, food toxins, radiation. But his concept was that there's also an internal environment that's directly related to the external environment. For example, obesity and chronic inflammation, which I'll talk about. And these together can lead to cancer biomarkers of risk and prognosis, and finally to cancer preventive and screening strategies. The cancer genome and epigenome, genetic susceptibility, of course, driver genes that are important in terms of targeting for therapy, but the understanding of carcinogens also feeds into this paradigm. Now, many cancers are caused by chemical agents, going way back to chimney sweeps and scrotal cancer with benzpyrene, or aflatoxin, which is from a toxin from a fungus, Aspergillus flavus, that causes liver cancer, and cigarette smoke. And it's more than 60 different chemical carcinogens that cause lung cancer and other cancer types. Now, in some cases, these specific carcinogens cause specific mutations in the p53 gene, which I'll talk about in more detail. Aflatoxin, liver cancer, G to T transversions at a particular site, 249 mutations, that was the very first one described by myself and others. And then polycyclic hydrocarbons, such as benzpyrene from smoking and lung cancer. And there are multiple hotspots of mutations, but they're all G to T with strand bias. Ultraviolet light, which was shown by Doug Brash, one of my postdocs, to cause CC to TT transitions, and particularly at these two sites. And more recently, this is a toxin that causes kidney failure and also kidney tumors and causes AT strand biases. Now, the most common mutated cancer in all mutated gene in all cancer, of course, is the P53 gene, which is shown here in blue. And for example, in ovarian cancer, it's close to 95%. So, of all the various genes that are mutated, P53 is the most common. And P53 is a tumor suppressor gene, but the mutants come in several different types. There are loss of function mutants, so they lose the ability to act as a tumor suppressor by multiple ways. But in some cases, these mutations lead to gain of function as an oncogene. So the P53 gene and its product changes from a tumor suppressor to an oncogene. And does this have consequences? The answer is yes. In multiple cancer types, gain of function P53 mutations are associated with poor prognosis, including lung cancer. So this is the line in red. This is patient survival, 100%, 0%. This is time in months, so this is a 5-year point. So there's a highly significant compared to the wild type P53 or the non-gain of function P53. And this is true in many, many different types of cancer. Now, as I mentioned earlier, one of the concepts that Chris Weil had with the expososome was that it was linked to infection and chronic inflammation. Now, chronic inflammation can be either largely inherited, such as hemochromatosis, an iron overload disease that causes liver cancer. Inflammatory bowel diseases is partially related to inheritance, and familiar pancreatitis and pancreatic cancers. So the former president, his family has high incidence of pancreatic cancer due to pancreatitis. Fortunately, he did not, still in his 90s, but he's actually doing pretty well with his metastatic melanoma at the point. But more frequently, the infection is inherited, acquired, such as viruses, hepatitis B and hepatitis C, or bacteria, such as Helicobacter, or parasitic infections, such as shown here. And there's about 2 million people per year that are related to various infections from the WHO report of a few years ago. But then there's another group of non-communicable diseases and conditions that increase the risk of cancer and also chronic inflammation. And these include acid reflux and esophageal cancer and Barrett's esophagus, obesity for multiple types of cancer, and smoking, which, as I said earlier, has about 60 different chemical carcinogens. It's a witch's brew of carcinogens, but it's also a very potent inflammatory agent. And anyone who's inhaled their first puff of a cigarette realizes how toxic it is. And if it wasn't for nicotine, I don't think anyone would be smoking cigarettes. So if you add these to non-communicable diseases, it's well over 6 million cancers per year worldwide. So there are various biomarkers that are associated with health disparity and increased risk. This is a study we did a few years ago. It was a prospective study in which we measured various inflammatory agents in the serum, and IL-8 and CRP, a C-reactive protein, were elevated even 5 to 8 years prior to the development of lung cancer and are various obvious risk predictors. At the time of cancer, serum IL-8 or IL-6, both in Afro-American and European Americans, are elevated, and these pro-inflammatory agents, such as interleukin-1 beta and the anti-inflammatory IL-10, in Afro Americans is elevated. And finally, in terms of survival and prognosis, high levels of IL-6 and IL-8 are associated with poor survival, and I'll show you that in a moment. Mannose-binding lectin-2, which is a genetic inheritance of a haplotype that leads to hyperreactive innate immunity, is associated with poor survival. TNF-alpha, pro-inflammatory agent, and anti-inflammatory cytokines 10 and 12 are in Afro-Americans. So there's a difference between Afro-Americans and European Americans in some of these risk factors that are primarily related to inflammation. So I mentioned that IL-6 and IL-8 were prognostic classifiers. In this case, a study we did a few years ago showing that even in the earliest stage of lung cancer, that's stage 1A, 3 centimeters or less, if neither one of these inflammatory cytokines are elevated, the prognosis is really quite good. If one out of the two is elevated, it's intermediate. But if both are elevated, then at a between 5 and 10 year period, 75% of the people are dead. 100% survival, 0% survival, 5 years, 10 years, 15. So even though there's a surgical intent of cure, there's a population of people who already have micrometastases. Now, passive smoking. This is the watercolor that was actually painted by Professor Hariyama, who was an epidemiologist from Japan. And there was a meeting in 1981 in Greece at Cape Sunion in which he presented his data for the very first time that passive smoking increased the risk of lung cancer. And at that time, not very many people believed him. But since then, there's been 50 to 80 different papers that have shown in multiple populations, multiple conditions, that passive smoking also is a risk factor. And I sort of like the flowery language of Hippocrates, in which he wrote, "Some men have constitutions that are like the wonderful, the wooded mountains running with springs, others like those with poor soil and little water, still others like land rich in pastures and marshes, and yet others with the dry earth of the plain." Even at this time, many, many, many centuries ago, physicians recognized that people were different from each other, and that they had some conditions that led them to have poor survival than others. So I was sitting at that meeting listening to Hariyama, and that was in 81. And I thought, well, I think I know the kind of study I want to do. And it was the following: to look at never smokers who develop lung cancer and ask the question whether or not their parents smoked. And but in 1981, it was much too early to do those studies, partly because women didn't start smoking until the Second World War with any frequency. And secondly, in most questionnaires, that was not asked whether or not your parents smoked. So I went back, actually came back here, and added those kinds of questions and others to our epidemiological case control studies. And many years later, Susan joined us for a couple of years, and this was 2009. And so we could test that hypothesis. And we found that childhood exposure to secondhand smoke and genetic conditions and innate immunity increase lung cancer risk in never smoking adults. And the smoking of either parent was important and additive, and even grandparents who took care of the kids, if they smoked, it contributed to the increased risk. And, and this was done in two different cohorts. So the conclusion was that that's a hypothesis. The conclusion was that parental secondhand smoke exposure during childhood is associated to a dose-dependent increase in lung cancer risk among never smokers in a Mayo Clinic cohort and one here at the NCI. And especially in those who had a mannose-binding lectin, a type 2 haplotype, and were hyper reactive innate immune system. And one of the surprising results that we found was that these never smokers who had exposure from their mom and their dad at an early age, they developed lung cancer in their 50s instead of their 60s or 70s. So in many kinds of studies, children are a susceptible population, and it's true in smoking-induced lung cancer. So now let's go to a genome. So this is a traditional view when Terry and I were initially doing lung cancer research, a small cell, adeno, and squamous. And then in 87, KRAS was found, an oncogene. And then in 2004, there were a number of other genes found, particularly EGFR. And I want to remind you that P53 mutations has been known throughout this time. And then 2014, a number of protein kinases were found to be mutated in some cases, such as ALK fusions. But these were only essentially in never smokers. So they're sort of misleading with this increase in these various genes because they rarely occur in smokers who develop lung cancer. Now, what was exciting at the time, and it's still very interesting, was that these could be targeted. So there were a number of different drugs that were developed, and and they're in the third or fourth generation of many of these drugs now. The satisfying thing was that there were complete responses in which many tumors just melted away. However, within a month or two, they came back. So they were quite temporary, and these drugs cost about $100,000, $150,000 a year. So it's an economic burden on these people, but that's the reality of the situation. Now, let me turn to the transcriptome, and first to microRNAs. So microRNAs are small non-coding RNAs that are evolutionarily conserved and regulate gene expression. And they control the protein output of hundreds of genes, which are repressed by each microRNA by either destabilizing the RNA or, to a lesser extent, inhibiting translation. And Victor Amaros, who eventually will win the Nobel Prize, discovered these in yeast in 1993, and then Gary found that that microRNAs were found in human cells and tissues. And then finally, Carlo Croce, who's pictured here, in 2004, found that microRNAs were differentially expressed in human cancers. And since then, it's been shown that microRNAs predict risk, diagnosis, prognosis, and therapeutic outcome. So as I mentioned, these very small RNAs, usually 24 to 26 base pairs, they bind to messages. They can inhibit translation of that message, but they can also cause instability of the message, especially if there's one or two base changes that are not that lead to the message interacting with the microRNA. MicroRNAs can also bind to proteins and alter protein function, and they can even act as ligands. For example, the toll receptor ligands that lead to inflammation through IL-6 and TNF-alpha. Well, we got started in this area when Nozumo joined the lab, and it was quite some time ago, 10 years ago now, and we found that microRNAs were significantly profiles different between primary lung cancers and corresponding non-cancerous tissues and among different histological types. So analyzing the microRNAs, you could determine that they were squamous cell or adeno or small cell. And we found that miR21 and 155 and 106 were increased, and there was a decrease in LET7, which is, if you want, a suppressor, tumor suppressor type of microRNA. That confirmed results from Japan, from my colleague Tak Takahashi and Frank Slack, who was at Yale at that time. And each of these were shown be associated with diagnosis and prognosis, including the earliest stage of lung cancer, stage 1. That led us to collaborate with Carlo Croce to ask a very simple kind of question at the time. Of the 6 major types of human cancer, which microRNAs that were known at that time, there were about 290 known at that time, there's well over a couple thousand known now, were most frequently overexpressed in these cancer types. And miR21 won the race in that in all these major cancer types, miR21 was the one that was most frequently overexpressed. And it was then shown by us and others that it was upregulated 18 major cancer types and was the biomarker of poor survival in at least 14 of those cancer types. So this is some data from our studies of lung cancer that increased miR21 expression in lung cancer is associated with poor prognosis in 3 different cohorts. This is very important when you're doing these kinds of studies to look at multiple cohorts. Maryland, in Baltimore, Norway, and Japan. So these are Kaplan-Meier plots again, 100% survival, 0% survival. This is the 5-year point. So above the median, high levels of miR21, poor prognosis, poor prognosis, poor prognosis. As I said, in 18 different cancer types, high levels of miR21 are associated with poor prognosis. And these are just some examples. These are studies from us, lung and colon, but pancreas, breast, head and neck, tongue, astrocytomas, leukemia, so on and so forth. Those are just examples in which high levels of miR21 are associated with poor prognosis. Well, what increases miR21 and what does miR21 do? It's amplified on chromosome 17q, and the latest version from TCGA, which is analyzing the genome of lung cancer that came out just a few months ago, has shown that one of the common features in lung cancer is amplification at this site, which is associated, of course, with miR21. There could be decreased transcriptional silencing in terms of DNA methylation that can, if it's decreased, can lead to an increase in miR21. We showed that EGFR mutations lead to an increase in miR21. And if you have an EGFR mutation, usually, again, that's an ever smoker, you don't have a KRAS mutation. If you have a KRAS mutation, you usually don't have an EGR mutation. And a number of inflammatory cytokines, such as IL-6, which I talked about just a moment ago, lead to an increase in STAT3. David Baltimore and his colleagues found that. And that STAT3 acts as a promoter, acts on the promoter of miR21, it's a transcription factor, to increase it. And then with some of the people here, we showed that either free radicals or ionizing radiation as a stress response. Well, what does miR21 do? It does many different things, has many different targets, and in each of these cases, it decreases the amount of these various proteins, which are in general involved in inhibiting processes related to cancer and cancer progression and metastases. Now, I mentioned that miR21 can also lead to changes in toll receptors. And this is a cartoon that I made from interesting studies that Carlo Croce has done, in which miR-21 can be in the cargo of exosomes, these small 100 nanometer really pockets of membrane-bound vesicles that then can fuse with immune cells. And when that happens, through the toll receptor, it leads to an NF-kappa B and the inflammation and increased tumor growth and metastatic potential. And then he found another interesting finding, which is being explored now in the clinic, that miR-21 can also, through these exosomes, lead to apoptosis of muscle cells. And this can be associated with cachexia. So I don't know how many patients that Terry took care of with cachexia, but I've taken care of a number of them. And they just melt away right in front of you. They're fat, they're muscles, and almost universally they die. So anti-MIRs against miR-21 might be of some value, and that's a hypothesis that's being explored now in South America, actually. So the transcriptome, that includes the microRNAs, but let's look at just the messages. And we found that there are 4 gene messages that are increased that are associated with poor survival. So XPO1 that transports microRNAs and other RNAs into the cytoplasm. BRCA1, you say, well, BRCA1, increased BRCA1, wouldn't that be a good thing? Because low levels of BRCA1 lead to a increased risk of breast cancer. But in fact, high levels of BRCA1 cause genomic instability. HIF1 alpha, high levels of that, you might expect that to be found in tumors that are hypoxic, but this is the message level. Hypoxia usually leads to a increase in the protein, the translation of the message, more than an increase in the message itself, and also a decrease in a known tumor suppressor gene that was discovered here by Nick Popescu a number of years ago. So these are all stage 1 lung cancers. Again, the cancer is less than 3 centimeters in size. In each of these cases, this 4-gene classifier was associated with poor prognosis. And then we looked at 4 new cohorts of stage 1 lung cancer and found that, in fact, in the literature, that the same observation was observed. So you can put all this together and look at odds ratios, and we prefer to use cancer-specific survival. The Japanese prefer relapse-free survival, and most people use overall survival, which isn't too bad in lung cancer, but overall survival doesn't take in consideration dying of cardiovascular events or anything else. But anyway, the increased risk associated with this was about odds ratio about 2, and this was in stage 1A lung cancer as well as 1B, when those are 5 centimeters and less in size. So again, mechanistic biomarkers, proof of principle in lung adenocarcinoma, the combination of protein coding genes and that are mechanistically related to lung cancer, adenocarcinoma, and non-coding genes. So these are the protein messages that code protein coding genes, and miR-21 is a better prognostic classifier than either alone. That's the hypothesis. Did it prove to be the case? And the answer is yes. So this is a Japanese cohort. So if the 4-gene signature was high, poor prognosis of miR-21 was higher. If the 2 together were high, then there was even a worse prognosis. And this is true also in the US-Norway cohort, in which there was higher frequency of 1B versus 1A in terms of stage, and again, high, poor prognosis, and then this is if neither are high, if one is high If both are high, again, very poor prognosis by either 10, 40 months, I guess. 75% of the people are dead, even with these early stage cancers. So what's the principle here? If you combine the non-coding RNA, for example, and encoding RNA, you get these kinds of curves. If you put them together, low risk, either one high, intermediate risk, both high, poor risk, they both give an agreement, then that gives you a better result. But why is there this better result that if they're mechanistically and statistically independent? Is that they each misclassify some individuals. So this is a coding RNA, and this is the misclassification with the non-coding RNA. So there's further proof of principle that taking two statistically and mechanistically independent biomarkers will give you a better result and in terms of prognosis, and that's for colon cancer, esophageal cancer, both adeno and squamous, lung adenocarcinoma. These are all studies from our group, and this is a study of breast cancer that Croce and others have done. So now let's turn to the epigenome. And when most people think of the epigenome, they think of DNA methylation. So this is Anna Robles in our group. And the hypothesis she investigated was an integrated biomarker classification of stage 1 lung adenocarcinoma based on independent message, microRNA, and DNA methylation biomarkers, which further improve the prognostic classification. So I showed you data previously. If you put two independent biomarkers together, statistically and mechanistically, that was better than either one. This is asked the question, if you put three, what happens? So the third one was DNA methylation, and there was already some evidence from the results from Manuel Esteller in Barcelona that DNA methylation at these 5 different sites was associated with a poor prognosis of stage 1 lung cancer. So we looked at these data and asked a very simple question. Are each of these sites of DNA methylation independent of each other, or are they all dependent on each other? So when we did multivariate analysis, we found that they were all dependent on each other. So HOXA9 was a little bit better than the rest, but the 5 wasn't better than HOXA9. So we just focused on HOXA9, and we put the 3 different biomarkers types together, miR21, the 4-gene signature, and the HOXA9 promoter methylation. And we published this, I guess, last year. So this, again, if all 3 are positive, very poor prognosis in stage 1 cancer. If 2 are positive, intermediate, 1 positive, much better. And if they were all negative, the prognosis in stage 1 lung cancer was almost 100%. And this is true in stage 1A, 1A, and in 1B. So now let's end up with the metabolome. We've done studies with the microbiome, but I don't have time to tell you about those studies today. And this is Maya and Evie. Evie's now at Ohio State. Maya's still in the laboratory. And the hypothesis is that unbiased metabolomics will discover biomarkers associated with risk, diagnosis, prognosis, therapeutic outcome of lung cancer. And we've utilized both serum and urine for measuring these metabolites and showed that some of these actually come from the tumors. And to go back to Hippocrates that I quoted earlier, he and other physicians actually tasted urine in the diagnosis of disease of his patients. He didn't know that that was associated with glucose and risk of diabetes, because diabetes hadn't been described. But he did know if he could taste a sweetness in the urine, it was associated with poor survival of the individual. So the physiologist Armstrong wrote, again in flowery language, from a liquid window through which physicians felt they could view the body's inner workings, urine led to the beginnings of laboratory medicine. So we've done studies of the metabolomics, metabolome. The first was published in 2014, in which we did unbiased analysis of metabolites that were found in the urine. And then we found that there were 4 that were associated with poor prognosis. So this is 100% survival, 0% survival. It's the 5-year point. And these are the 4: creatine riboside, N-acetyl-neurominic acid, cortisol sulfate, and a lipid that we're still working in terms of identifying. And we found that all of these are also found in the tumor. So if all 4 of these were detected at a high level, the prognosis was quite poor, and only about 5–10% of the people were still alive if it was that in their urine all 4 of these metabolites were found. So are they from the tumor themselves? And so we analyzed tumor and the matched non-tumor region from a series of lung cancers, and we found that the creatine riboside was quite high. And there was a positive correlation between the amount of the riboside in the tumors and the amount in the urine. So we then asked about risk. So can you measure these metabolites in urine of people in a prospective study to ask the question, if they're high, are these people at increased risk of developing lung cancer? So we just published this quite recently, and this is the kinds of analysis that were done, and that the multivariate analysis for these 2 were associated with an increased risk. And if you do what's called a ROC curve, that's looking at sensitivity and specificity. And this is if it was like 0. But if you put these 2 together, the sensitivity and specificity and the area under the curve was actually quite high, and the predictive value, negative, positive predictive value, and negative predictive value, and correctly classified, was respectable at 75%. And this is the prospective aspect of it. This is from a study that was started more than 30 years ago by the Division of Cancer Epidemiology at that time. And so urine was collected, and actually this was serum collected, and we looked at IL-8 and C-reactive protein, and found that if the 2 together, let's just look down here, if the 2 together were found that it was associated with an increased risk of lung cancer. Now let's go back to the metabolomics again. And so we asked the question, is there a difference between European Americans and Afro-Americans? Health disparity, if you will. So if the high levels of creatine riboside and NANA were both found, then the odds ratio in multivariate analysis was quite high, above 10 for each one of these. And that's actually quite an important value because in terms of a risk test. But in Afro-Americans, there was not this difference. So this showed that there were some significant differences between Afro-Americans and European Americans. And if you do the ROC curve again, this is sensitivity and specificity. The 2 together is the green line here, and the area in the curve is quite good, and this is the specificity non-negative predictive value, positive predictive value, and 84% of these people were properly classified. So what does that mean? That means that from a clinical standpoint, one of the questions we have as physicians is, what do we do next? Do we do watchful waiting and keep doing whatever screening test we're going to be doing, or do we, in some cases, go ahead and do surgery or biopsy if there's maybe a lesion there? And so if there's a high negative predictive value, that means it's unlikely that that person is going to develop cancer. So watchful waiting is one choice that one has. Now let's end up with more or less where I started, and that's low-dose CT. So low-dose CT was tested over a number of years and was published in 2011 in New England Journal of Medicine. And there were two populations, one that got, in quite large, 26,000, that got chest X-ray, which was sort of the standard of care at that point, or of diagnosis at that point, even though most people thought it was a waste of money, and a low-dose CT, that's computerized tomography. And they found that after the randomization, that those people who had a chest X-ray, they had a poorer survival than those who had low-dose CT. And the tumors that were detected were primarily these very small tumors that I've been talking about, stage 1A. But there was a problem, and the problem is a false positive rate of 96%. So that meant that most of the nodules that were detected by low-dose CT were not cancer at all. They were benign lesions. And this is a visualization of what this might look like. So this is you say the Verizon Center, for example, and these are seats of people who actually developed lung cancer that were tested over 3 tests, actually. But these are seats, the dark areas here are seats of false positive. So you have a sea of false positives and a underrepresentation of those that actually have lung cancer. And so what do you do with these people who have these lesions? Maybe you develop biomarkers that will predict ones which are more likely to be cancer versus those which are not. And to do a biopsy, and only about 2/3 of the lesions, or maybe 50%, could be reached through a bronchoscope. Or you do surgery and you remove them. So there's a lot of overdiagnosis. In fact, in the US right now, using low-dose CT, over 50% of the patients who have the lesions removed have benign tumors. And it's not a small problem to do surgery and open someone's, as the surgeons say, crack someone's chest. Well, low-dose CT is now the standard, and it's actually quite rapid that both the Affordable Care Act, assuming that's still in place in the near future, and also Medicare mandate coverage of low-dose CT. So these people that are eligible or high risk between 55 and 74, a 30-pack year history, quit within 15 years of current smoking, and we need to identify high-risk individuals outside this criteria. About 10% of the people coming to our clinics now are never smokers. Maybe they were exposed to the smoke of their parents, or they were passive smokers, or they there was radon or asbestos or something else. So there's a need to identify people with the criteria to prioritize those at greatest risk. I have 8.8 million. It's been upgraded to about 10 million people now. So there's no way that we can do low-dose CT on 10 million people or even 1 million people. There's just not the capacity to do that as yet. That's changing because there's money to be made and maybe people's lives to be saved by a low-dose CT. So there's a need for prioritization. And that goes back to this last point. And this is a cartoon from a review that we, actually two reviews, one in Nature Reviews Cancer that came out a couple of months ago, another in lung cancer that you can read if you're interested in lung cancer precision medicine. So let's just summarize this. So the goal of precision medicine to identify 25% or so of the stage 1 lung cancers whose cancer will recur because they already have metastases, micrometastases. It's about 25% of the stage 1 lung cancers, maybe a little bit higher, will recur and die of the disease. So can we use precision medicine and the information commons to decrease the false positive rate, decrease financial cost, improve patient care, and guide mechanistic studies? So people could be either low risk of recurrence or high risk of recurrence with either stage 1A or stage 1B after surgery. People who are at low risk observe, watch for waiting, and getting further tests. But people who are at high risk of having a metastases, a micrometastases, maybe adjuvant immunotherapy or chemotherapy or both. And there's a hypothesis which I think needs to be tested, and that is micrometastases may be more sensitive to traditional therapies or immunotherapy than when you actually have a metastases, which is a tumor of several centimeters or even more. So that's the message I wanted to leave you with. And these are my colleagues. A number of them have their own laboratories now. Some are still here, such as Anna Robles and Bree Ryan, and they've been replaced by others. And then these are our collaborators from other institutions. And that's it. Thank you for your attention. Yes, please. Yes. Okay. So the question is, how do you integrate various biomarkers from the various omics, whether it's genome or epigenome or transcriptome or metabolome or microbiome? It requires biocomputing. It involve big data. But the key thing is to show that each of the biomarkers is independent of the others, statistically independent, and hopefully mechanistically independent. And that's why simple tests like multivariate analysis is of some value. So to better analyze these different biomarkers, that's an area of advanced computing that's going on right now, and being with some success. Other questions?
What about the chest X-ray? Do you think they should do away with that?
Yes, I think the chest X-ray has been proven time again that it has essentially no value. In fact, it probably has a negative value. By the time you see a lesion on a chest X-ray, it's usually already metastatic, and the patient's going to die anyway. And the kinds of therapies we have are not very effective, although there's once again hope for immunotherapy of lung cancer, and about 20% of the people appear to have enduring, long-lasting, complete responses. So that's much better than we've done in the past with chemotherapy or with radiation therapy. So I think chest X-rays are a waste of money. They expose people to radiation, and they're rapidly being replaced by low-dose CT.
All right, that'll do it.
We're done. All right.
You all got an email today about the final exam. So for those of you who want a certificate, you have to take and pass the final exam, answer 70% of the questions right. There's one question from each of the lectures. It's open book, open notes. And when you take the exam, you have to finish your work. You can't save it. And then once you pass the exam, you fill out a course evaluation. And you have up till the middle of January to take the exam. So there's no rush. Enjoy the holidays. And our next speaker is Marina Dobrovolskaya, and she was educated in Russia. She then came to the U.S., was at the University of Maryland, Baltimore, and then in Virginia. And she's been at NCI now for almost 10 years. And she's a senior principal scientist, the immunology section head of the Nanotechnology Characterization Lab up in Frederick. Her title, Nanotechnology for Cancer Therapy, Benefits, Concerns, and Effects on the Immune System. Marina.
Thank you. Does everybody hear me? Is it okay? Good evening, everyone. Hi to everybody in this room and to those of you who is watching this presentation online. I'm going to talk about nanotechnology for cancer therapy. Obviously, it's a huge area. I'm presenting every year, so this year I decided to change the topic a little bit and restructured it so that I will give you some general overview of the field. We'll talk about the definitions for nanotechnology. I will give you some examples of the nanoparticles used in daily life. Then we will look at some examples of the nanoparticle application in medicine. And the main body of my presentation is dedicated to cancer diagnosis and therapy. I will use some examples to show you how nanotechnology can benefit cancer therapy. We will also discuss some of the safety concerns that are associated with using nanotechnology carriers. And because my expertise is in immunology as it relates to nanoparticles, I will try to focus these discussions to use immunotoxicity and some elements of how particles can interact with the immune system. And in this interaction, I would like you to remember and carry it for the end of my presentation, one thing, and that is particles can be toxic to the immune system, but their effects on the immune system can be engineered to benefit therapeutic applications. So there is like a double edge effect, right? So they can be bad or they can be good. And I think the goal of the nanotechnology is understand what is causing undesirable effects and turn it to benefit therapeutic applications. So what is nanotechnology? There is no universal definition. Often, many people refer to the definition by the National Nanotechnology Initiative that defines nanoparticles as objects with size and at least one dimension from 1 to 100 nanometers. According to the US Food and Drug Administration, even if size dimension falls outside of the nanoscale range up to 1 micrometer, so which is 1,000 nanometers, if the product is engineered to exhibit properties or phenomena related to their size, the FDA would consider it as a nanotechnology. So basically, if you look at the scale of size of different objects, then we talk about nanotechnology, we talk about the objects with size ranging from the size of molecule of glucose to the size of bacterial cells. There are a lot of nanoparticles in our daily life. Often we use some products without realizing that they contain nanotechnology. For example, according to the literature survey, there are more than 800 manufacturer-identified products currently on the market that contain nanotechnology. They include wound dressing, clothing, washing machine liner, coating for the lenses and sunglasses, some sporting equipment. In sporting equipment, mainly nanoparticles are used due to their antibacterial property to prevent transmission of certain microbial associated diseases from one user to another. All of us like summer, right? And raise your hand if you like going on the beach during summer, a nice summer day, like I do. And what we do, we have to use sunscreens, right? Because we need to protect us from the sunburn. So did you realize that if you use sunscreen that doesn't leave the white mark on your skin, this sunscreen contains nanoparticles? Usually all translucent sunscreens contain nano-sized titanium oxide or zinc oxide particles. The liposomes and emulsions are commonly used in cosmetics. So if you see super expensive anti-wrinkle cream from L'Oreal, you know that they contain nanotechnology and specifically liposomes and emulsions. Carbon nanotubes are often used as structural materials due to their durability. This is the summary from a study published recently by Mark Etheridge. So what he tried to do, he surveyed the literature to understand what are the common features of the nanoparticles used in medical applications. What you see here, that most nanotechnology formulated drugs and devices are intended for cancer therapy. So their primary market is cancer therapy. Most of them are intended for systemic intravenous administration. And most of them have size less than 350 nanometers. So very common sizes are 100 and 200 nanometers. Most of these materials are also neutral, have hydrophilic surfaces, and have spherical shape. Why it is important? Later in my presentation, I will tell you about the role of the physicochemical properties of the nanoparticles and their interaction with immune cells, with biological systems. And you will see how the shape, size of the particle, and the surface charge affect their immunocompatibility, biocompatibility, and toxicity of these materials. Same study also investigated the difference in the type of nanomaterials that are present in investigational nanotechnology-based drugs and devices and those products that are currently on the market. And what you see is that the dominant material in the investigational field is liposome, followed by polymeric nanoparticles, followed by emulsions, followed by solid lipid nanoparticles. Why it is important? Because likely the materials that are currently in investigational stage, this is what we will see being translated to the medical applications in the next 5, 10, 15 years. Dominant type of nanomaterial that is currently in use, in the commercial use, is solid nanoparticle, followed by nanocomposites. But you see that there are other materials, including protein nanoparticles, polymeric nanoparticles, emulsions, virus-like particles, nanocomposites, etc. Nanotechnology has a lot of benefits to offer to drug delivery. When it comes to cancer therapy, many of you who do research in oncology know that there are plenty, hundreds of the small molecule cytotoxic drugs that are very potent in killing cancer cells in vitro. But one very common problem of these drugs is they are not soluble in water. So we cannot use insoluble, we can dissolve them in DMSO and inject into animals to show that they are toxic or to treat cells in vitro. But we cannot use DMSO in human patients. So one of the benefits of nanotechnology is to overcome insolubility of the small molecular drug. Nanoparticles can also offer multifunctional capability. By multifunctional, I refer to the particle that carries a drug, which could be small molecule or biotechnology-derived therapeutic, but it can also carry an imaging agent. It can also carry a targeting moiety on the particle surface. Aptamers, antibodies, protein, and peptide ligands are commonly used as a targeting moieties, and we will talk about targeting in the next slide. There are certain robotic tasks such as sensing, computation, and triggered responses that can also be mediated by nanomaterials. I hope I intrigued you by tumor targeting. So let's see what tumor targeting means. Then we talk about targeted delivery of therapeutics. In terms of the nanotechnology for cancer therapy, we are talking about two primary effects. One of them is called passive targeting, which is based on the so-called enhanced permeability and retention, EPR effect. And the second one is active targeting. So the passive targeting is based on the phenomenon which is known that due to their rapid formation, blood vessels around the tumors are leaky. So that being small, nanoparticles can easily penetrate through the endothelial barrier and go into the interstitial space. But because they are larger than small molecules, they do not wash out of the interstitial space. So they enter the tumor space and then they stay around the tumor area, hence this terminology, enhanced permeability and retention. So for cancer therapy to work, nanoparticle has to enter the tumor space, release the drug, and make this drug available to the cancer cells. Then we talk about active targeting. We talk about the addition of a certain moiety to the nanoparticle surface, which makes this particle specific to cancer cell. For example, cancer cells could express cancer-specific antigens on the surface. And then we use antibody or ligand that specifically binds to that antigen. We can enhance delivery of the anti-cancer drug using nanoparticle to that cancer cell. However, it is very important to keep in mind that in order for the active targeting to work, passive targeting has to take place. So before the targeting ligand has a chance to show that it is working and it's worth of using this targeting ligand, particle has to cross endothelial barrier and go into the interstitial space to get in touch with the tumor that has a certain antigen. Another benefit from nanotechnology is a reduction in the drug toxicity. And there are many examples. Since, again, I mentioned to you that my primary expertise is in immunology, I will use immunotoxicity as example, and I will show you two drugs. One drug belongs to the small molecule class, and the other one is therapeutic protein. And what I'm doing here, I am comparing traditional formulation versus nanotechnology formulation. So let's see at the small molecule. Many of you know anti-cancer drug doxorubicin. And many of you know that doxorubicin main toxicity is cardiac toxicity. And I will talk about it in my next slide. But for the purpose of this slide, I would like to bring another toxicity of doxorubicin, which is induction of DIC, which stands for the disseminated intravascular coagulation. It is a very common dose-limiting toxicity in therapy of the especially acute myeloid leukemia. Then patients are given doxorubicin. It induces procoagulant activity complex, PCA, on the surface of the cancer cells that trigger blood coagulation. And therefore, in order to save patient, this therapy has to be terminated. Then the same drug is formulated using pegylated liposome. This formulation is known as Doxil. Nanotechnology formulation of doxorubicin does not have DIC and associated with this procoagulant activity, which is believed due to the prevention of the rapid release and contact of the drug with the cancer cells. My next example is from therapeutic proteins. Tumor necrosis factor alpha is a cytokine. It's a therapeutic cytokine that was tested in clinical trials, in fact, here at NIH 10-15 years ago. And these clinical trials failed due to the systemic immunostimulation, which makes sense, right? Because it's a cytokine. What cytokine does? It induces immune cells, and it is delivered into the blood into the quantities sufficient to kill cancer cells. It would also result in overwhelming immunostimulation. A small biotechnology company here in Rockville, Maryland, Cytimmune Sciences, formulated TNF on the surface of the pegylated colloidal gold. And also the clinical trials phase 1 was conducted here at the NIH. I had the privilege to witness one of the patients receiving this therapy. This formulation successfully passed clinical trials because there was no overt immunostimulation. And this is believed due to the masking of the TNF on the particle surface by the polyethylene glycol coating. Very hot topic these days is immunotherapy. Nanoparticles can be used to deliver immunotherapeutics, but I would like to draw your attention to another area of application of nanotechnology where nanoparticles are used to deliver cytotoxic drugs, and by this to potentiate efficacy of traditional immunotherapeutics. This is one of the very recent studies published by MedImmune. What they show, they compare two drugs, doxorubicin and pegylated liposomal doxorubicin doxil, in two models. At certain dose, which is in this case was 5 milligram of the drug per kilogram, if they use CT26, which is colorectal cancer model, in athymic nude mice, they do not see a difference between the formulations. It's not efficacious. You see that the tumor keep growing. However, if they use the same drugs at the same dose in the bulb C mice, which are immunocompetent mice, and that CT29 is the orthotopic tumor for this model, all of a sudden they see that Doxil has some effect. The tumor still grows, but the growth of this tumor is delayed. Many of you may have heard this terminology. It's one of the very recent terminology, immunogenic cell death. So the anthracycline antibiotics, which is like doxorubicin is, are known inducer of so-called immunogenic cell death. What is it? So the immunogenic cell death is basically the induction of the cell death that is accompanied by release of so-called danger signals. Okay, so the purpose of the danger signals is to attract immune cells, to activate these immune cells, and increase their natural response to cancer. So this study hypothesized that Doxil has the ability to enhance the immune response. And so what they did next, they combined Doxil with traditional immunotherapies. And what you see on this table, I prepared this table based on the data presented in the study. If they use Doxil alone and then traditional immunotherapeutics, anti-PD-1, anti-CTLA-4, and this is OX40 ligand, which is, it's not approved yet for clinical use. It is still in the investigational stage. And they compare the tumor shrinkage. So what you see here is a number of animals which responded to this treatment. So you see the Doxil, out of 12 animals, only in one animal there was some effect from Doxil. There was 4 animals, 2, and then 0 animals in PD-1, CTLA-4, and OX40 ligand respectively. However, when these therapeutics were combined with Doxil, the number of animals who successfully responded to therapy increased substantially. So Doxil can improve efficacy of cancer immunotherapeutics. It was shown in this CT26 mouse model of colorectal cancer. The mechanism is not yet completely understood, but there is a lot of research going on these days in this area. Next example is Abraxane. So Abraxane is a nanotechnology formulation of the anti-cancer drug paclitaxel. Okay, so this is Abraxane is a paclitaxel formulated using albumin nanoparticles. And combination of Abraxane and anti-PD-1 is currently in the clinical trials for the metastatic breast cancer. Again, nobody yet understood the mechanism completely, but idea behind it is the same as I described you for the Doxil. Is the purpose of the anti-cancer drug is to do the damage to the tumor, do the damage to the blood vessels around the tumor, and the nanotechnology carrier can induce some inflammation and promote the immune response, the normal immune response against the tumor. Another benefit is using nanoparticles for vaccines. Nanotechnology can benefit a vaccine efficacy not only in cancer therapy, but developing cancer vaccine is especially difficult. And I think this, there is no, to my knowledge, there is no clinical formulation yet, but I know a lot of examples of using nanotechnology for cancer vaccines that are either in a preclinical stage or in clinical trials. This is one example from one of the very recent studies a few months ago published in Nature, where what they did, and I encourage you for more details to read the actual paper. So what they did, they took the mRNA encoding the tumor-specific antigen, and they formulated this RNA using lipid cationic lipids to form so-called lipoplexes. And so the purpose of the lipoplexes was dual. One, lipoplex protected RNA from degradation by nucleases. And second, lipoplex, because lipids are yummy, immune cells love lipid particles. So if you don't protect the surface of the lipid particles with hydrophilic coating like polyethylene glycol, immune cells will grab it and engulf it. So lipoplex induced uptake by RNA, and also because RNA, our body has natural response to RNA-based viruses, it also built the immune response against RNA, and this immune response is manifested by production of interferons. So basically, what they showed, and they draw this cartoon, that they do systemic delivery, IV delivery of lipoplexes. These nanoparticles go into the resident lymph organs, such as spleen and resident lymph nodes, where it induces two waves of type 1 interferon. First wave is very quick, which is induced in the plasmacytoid dendritic cell. Second wave is delayed response. This is in tissue-resident macrophages. And it induces, the lipoplex induces maturation of the dendritic cells. Cumulatively, as a result, it culminates with induction of strong effector and memory T cells. And they show a lot of examples from the preclinical studies in melanoma models in animals. And this is the data from the studies, from actually clinical portion of the study, where they show the increase in the type 1 interferon levels in the plasma of 3 patients injected with this material, and the induction and maintenance of the cytotoxic T cells, CD8 positive T cells that are specific to the tumor. Since we talk about benefit of vaccine, I would like to mention another benefit of nanotechnology, which is delivery of the drugs and delivery of the nanoparticles and antigens or drugs associated with them into the lymphatic system. It is important for vaccines, but it is also important for therapy of disorders which are mediated by pathogens such as HIV or such as some parasites and intracellular bacteria that escape immune response by hiding in macrophages. And macrophages are residing in lymphatic system. This is example from the study published by the Jeffs Hubble lab several years ago. What they do, they show the role of the particle size in the lymphatic delivery. They use polymeric particles with 2 sizes, 100 nanometers and 25 nanometers. And these particles had a fluorescent label. After intradermal injection, you see that 100 nanometers particles all stayed at the site of injection, and they did not distribute to the lymph nodes. But 25 nanometer particles rapidly traveled through the lymphatic, this is tail, and then they were found in the draining lymph nodes. So the take-home message from this is, if we want to stimulate immune cells, antigen-presenting cells at the site of injection, like traditional adjuvants do, then we use large particles. But if we want to promote immune response and achieve sustained immune response, we would benefit from delivery to lymphatic. And we can achieve so by changing size of the nanoparticles to reduce their size to below 100 nanometers. Often the benefits of nanotechnology are hampered by concerns about safety of these novel materials. I would like to share with you 2 examples, but before we talk about these examples, I would like you to realize and remember 2 things. Nanotechnology carrier, nanoparticle, can be toxic. And API, active pharmaceutical ingredient, can also be toxic, right? Both the carrier and the drug can be toxic. In some cases, nanoparticles may reduce toxicity of the drug, which is some examples that I showed you as the benefits. But there are some examples, if we don't understand the property of the drug and the carrier, some nanoparticles can exaggerate or enhance toxicity of the drug. And this one of the examples is shown on this slide. Therapeutic oligonucleotides are often cannot be used in human patients due to induction of interferons. We also know that lipid carriers are pro-inflammatory and induce cytokines. So if we use lipid carriers for delivery of therapeutic oligonucleotides, we will have a product which combines both responses, which is not good, right? So this is example from one of the studies that we conducted at the NCL. This was therapeutic oligonucleotide. You see that nanotechnology platform did not induce interferon, but the drug, oligonucleotide, induced interferon. The final product induced interferon. But the platform induced cytokine. API did not induce cytokine. However, the final product induced the cytokine. So we have the final product which induces both interferons and cytokines. Therefore, immunostimulation and immunostimulation-mediated toxicity of this drug is enhanced. And this product went through the phase 1 clinical trial. However, it was discontinued and removed from the company's pipeline due to the overt immunostimulation as one of the reasons. There were other reasons for discontinuation of this product. We also talked about the so we talked about the drug toxicity and the particle toxicity. Another interesting element is that nanotechnology carrier can alter toxicity of the drug by changing biodistribution of the drug. And I would like to use example of the doxorubicin again. So we know that doxorubicin accumulates in bone marrow and heart, and as such, the common toxicity of this drug is myelosuppression and cardiac toxicity. Doxil is a pegylated liposomal formulation of the same drug, and benefit of the doxil is reduced cardiac toxicity. So it's a victory in one area. But problem in another area, liposomal doxorubicin now accumulates in the skin instead of accumulating in the cardiac tissue. And as such, there is a new toxicity, palmar-plantar erythrodysesthesia, also known as hand and foot syndrome that you see appears here on this picture as a rash on the patient's hands. This example is from research formulation. It was not used in clinic. It was only used in animals, where doxorubicin was formulated using cyanoacrylate nanoparticles. The cardiac toxicity was not an issue. Myelosuppression was not an issue. PPE was not an issue, which the authors of this paper celebrated by saying that, well, we finally achieved reduction of all of these undesirable toxicities. However, unfortunately, they also found that due to the small size of the cyanoacrylate nanoparticles, they distributed to the kidney, and therefore toxicity of doxorubicin traveled to the kidney, and this formulation was nephrotoxic. So API toxicity can relocate, which depends on the particle biodistribution, and this is an important factor that we need to keep in mind. And now let us talk about nanoparticle interaction with the immune system. For the purpose of this presentation, I selected several areas which are common, either common toxicities experienced with nanotechnology carriers and nanotechnology formulated drugs, or represent the areas of especially special interest in the research community. So we will talk about plasma proteins and how plasma protein binding to the nanoparticle surface affects particle biodistribution and uptake by the mononuclear phagocytic system. We will talk about particle effects on erythrocytes. We will talk about blood coagulation system, and I will use some of the examples of how particles can be toxic to the platelets, leukocytes, and endothelial cells. We will talk about allergy. The reason I chose allergy is because there is an increasing interest in this area from many researchers, both from environmental occupational safety standpoint, but also from the therapeutic application of the nanomaterials. We'll also talk about cytokines and immunogenicity. So proteins and nanoparticles. There is a mutual interaction between the proteins and the nanoparticles. Proteins may interfere with nanoparticle targeting, may affect dissolution of nanoparticles and change their physicochemical properties, and I will show you some examples in my next slide. They may influence particle biodistribution through opsonization and increased uptake to the organs of mononuclear phagocytic systems. Nanoparticles, on the other hand, can change the protein function. They may change either increase or decrease activity of the protein, change protein stability, or change protein conformation, which results in exposure of the protein epitopes, which are otherwise unavailable to the immune system. So this is an example from the study done by Tommy Sederwal, who showed how particle size influences protein binding. So he shows here that particles with a size 5, 10, 25, and 50 nanometers relevant to the size of the common plasma proteins, the high-density lipoprotein and serum albumin. And you see that the larger the particle, the greater the surface for the contact surface from the particle to bind to the particle surface. This is the example that shows the opposite interaction, how protein binding now affects the particle size. This study was done by us at the Nanotechnology Characterization Lab. Here we use colloidal gold nanoparticles with a nominal size 30 nanometers, and we use dynamic light scattering to measure this size. So you see that dynamic light scattering shows us 33 nanometers, which is very close to the nominal size of the particle. Look what happens after this particle is incubated with human plasma. The particle size almost doubles. From 33, now it became 76 nanometers. And this is because now this particle is surrounded by the protein corona, these proteins that are bound to the particle surface that increases hydrodynamic size. Why do we care about protein binding? Because it affects particle biodistribution, right? There are a lot of proteins in plasma that are called opsonins. Opsonins, and opsonization means binding of opsonins, and opsonins are proteins that improve or enhance uptake by the phagocytic cells. This is example from the study that we conducted in collaboration with Cytimmune Sciences. This is a schematic representation of the colloidal gold nanoparticle, and this is colloidal gold nanoparticle coated with a polyethylene glycol. So uncoated particles are anionic, so they can interact with plasma proteins, and you can visualize these proteins by two-dimensional gel electrophoresis. Particles that bind a lot of protein tend to accumulate very quickly in cells such as macrophages, and here I show you the transmission electron microscopy image of the macrophages. These black dots are gold nanoparticles, and in vivo they are taken up very quickly from circulation by liver and spleen, and you see that the color of these organs changes, right? But if we change the particle surface to create hydrophilic shield on the particle surface, the number of the proteins binding to the particle reduces significantly. These particles are not taken up by macrophages in vitro, and they are not eliminated quickly by the MPS organs in vivo. So there is a good correlation between in vitro and in vivo. Then when it comes to the MPS uptake, I would like to share with you current state of the knowledge in this area. And this is, as models. These are the models that were proposed and effectively researched by William Zamboni in the University of North Carolina. So Dr. Zamboni proposed two models of the nanoparticles captured by the MPS. One model is called capture, and the other model is called hijacking. So in the capture model, nanoparticle travels with the blood to the tumor and other tissues, liver and spleen, and is taken up by resident macrophages or phagocytic cells at in these organs. In the hijacking system, particle is taken up by monocytes in the bloodstream, and then monocytes deliver this particle to the inflamed tissues, to tumor, to the MPS. This model is now under active investigation because a lot of researchers consider this model to deliver drugs using nanoparticles. In other words, instead of trying to struggle with targeting nanoparticle surface and waiting for the nanoparticle to cross endothelial barrier into the tumor space, the idea is, what if we deliver we inject the nanoparticle systemically, and monocytes will take up this particle and then deliver the drug carried by nanoparticle into the tissues where it is required. There are a lot of concerns and benefits from using this model. I will skip it for the sake of time, but if you would like to know more, please refer to the studies by William Zamboni from University of North Carolina. Now let us talk about some hematological toxicities and nanoparticle effects on the immune cell functions. Erythrocytes are the dominant cells in blood, and materials that are injected systemically can benefit erythrocytes. For example, perfluorocarbon nanoparticles are used to stabilize erythrocyte membrane, and in fact, they are used like artificial blood. But some nanoparticles may damage erythrocytes, and erythrocyte damage is accompanied by some systemic and vascular effects which are listed here, and they depend on the particle size, and also they depend on the nanoparticle surface charge. This is example of how nanoparticle physicochemical properties influence the interaction with erythrocytes. The study was conducted in our lab many years ago. As a model particle, we use PAMAM dendrimers. These dendrimers are dendrimers are polymeric nanomaterials that have a globular spherical shape. They range in size. G refers to the generation. The higher the G number, the higher the generation, the larger the particle is. So we used particles with generation 3 and generation 6, and for each generation we had different terminal groups. We had succinamic acid, which is anionic, amine, which is cationic, and ethanolamine, which is a neutral particle. And this is what we observe. If we take particles with the same surface charge, amines in this case, if the particle is small, it does not damage erythrocyte. However, if the size of the particle increases, which means that the density of the surface group is also increases, then we see the hemolysis. The surface charge is also important because now we take generation 6 particles and we compare amine surface to carboxy and hydroxy surface. And you see that only amine-terminated particles, but not their anionic and neutral counterparts, damage erythrocytes. Coagulation system is another system that is commonly affected by nanomaterials after their systemic administration. Here, I would like you to remember two things. One is that coagulation system is a constant balance between procoagulant and anticoagulant activity. Shift in the balance in either direction results in either thrombosis or hemorrhage, and neither one is good. And second fact is that this is multi-component system. It includes platelets, it includes endothelial cells, leukocytes, cancer cells can contribute to the thrombogenic properties. Nanotechnology can benefit coagulation system, but the nanoparticles can also be toxic. Here, since I'm talking about toxicity, I will show you only the negative effect. If any of you is interested about the beneficial effects of the nanoparticles for coagulation system, please refer to this book, Handbook of Immunological Properties of Engineered Nanomaterials. So undesirable effects of nanoparticles on coagulation system includes interaction with coagulation factors, which can result in contact activation of these coagulation factors, binding and depletion of these coagulation factors. Nanoparticles can be cytotoxic to endothelial cells. They can also induce inflammation and oxidative stress, which will lead to the thrombogenic effects. Particles can also affect platelets and cause either direct or indirect activation of the platelets. Here, I show you examples how particle physicochemical properties interact with various components of the coagulation systems. Remember, when we talked about hemolysis, I showed you the structure of these PAMAM dendrimers. The same PAMAM dendrimers were used in this study, and the effects were very similar. So if we take small particle, this generation 3 dendrimer, it does not induce platelet activation. If we use the particle with the same terminal groups but larger in size, this generation 6, we see strong activation of the platelets, very similar to the positive control. These are the platelets in control sample, untreated. You see, when they are inactive, they present as a single cells. However, these are the particles activated by generation 6 PAMAM dendrimer with amines on the surface. We cannot detect single platelets because we observe these clumps of the cells. The terminal groups are very important. Similar to hemolysis study, we observed that particles with the same size but different surface functionalities have a different ability to activate platelets. Amine-terminated particles induce platelet aggregation, while their neutral and anionic counterparts do not. The density of the surface groups are also important. This is the effect of the platelet of the amine-terminated dendrimers on the platelet aggregation. And what we did, our chemists masked 25% of amines on the particle surface, and we observed slight decrease in their thrombogenic properties. then another 25% will mask, and another 25%, and another 25%. And as we keep masking the amines on the particle surface, we decrease the particle's ability to activate the platelets. This is another good example to demonstrate that by modifying surface properties of the nanoparticles, we can change their interaction with biological systems. Particle composition is also important. Here I am comparing platelet aggregation in response to PAMAM dendrimers and triazine dendrimers. This is triazine dendrimers, this is PAMAM dendrimer. In both types of materials, we see the same trends that we observed before. The larger the particle, the greater the effects on the platelets. However, you can also see here that at a similar generation and a similar concentration, PAMAM dendrimers, which have a higher number of the terminal groups, are more active than the triazine dendrimers with the same size. As I mentioned to you, the coagulation system also includes leukocytes, and leukocytes can contribute to coagulation by expression of so-called procoagulant activity complex, or PCA. This procoagulant activity complex is composed of two molecules, a phospholipid, phosphatidylserine, and the protein, tissue factor. And then this complex is present on the cell surface. It serves as a platform for attaching of the coagulation factor and initiates plasma coagulation cascade. What I show you here in terms of the leukocytes also applies to the cancer cells, also applies to the endothelial cells. Again, I'm using here example of the dendrimers. You see that the larger the particle, the more potent it is in inducing procoagulant activity. And also the surface charge is important. Again, cationic materials are more toxic than anionic and the anionic and neutral counterparts. Remember at the beginning I told you the most nanomedicines that are currently in development and in clinical trials are neutral. And this is why they are neutral, because neutral means less off-target effects. However, cationic charge is needed by chemists to conjugate drugs to the particle surface. And cationic charge is also important for delivery of certain drugs, such as therapeutic nucleic acids, because of the electrostatic complexation between the nucleic acid and the cationic platform. So the take-home message from these studies is that since cationic charge is toxic, right, it damages erythrocytes, it results in changes in coagulation profile in the multiple components of the coagulation system. One has to find the optimal balance between the number of the surface amines that you can still use to attach the drug and complex the nucleic acid, but which would be non-toxic when this material, the final product, is delivered systemically. This is a summary presented by Dr. Jan Simak from the FDA about nanomaterial effects on endothelial cells. So the nanoparticles can have direct toxicity to endothelial cells, and they can indirectly activate endothelial cells through induction of hemolysis, activation of the white blood cells, platelets, plasma coagulation system, and complement system. And as a result, when endothelial cell is dysfunction, the endothelial cells would die. And death of endothelial cells is associated with severe effects, which could be, depending on the magnitude, can result in systemic toxicity. Now let us switch a little bit the subject and talk about allergic reactions to nanomaterials. As you know, there are four main types of allergies. Type 1 of allergy is known as immediate type hypersensitivity. It is mediated by immunoglobulins E. Type 2 and type 3 immune hypersensitivity reactions are immune complex and cytotoxic hypersensitivities. And type 4 reaction is delayed type hypersensitivity, which is mediated by the antigen or allergen specific T cells. There is also another type of allergy called pseudoallergy. The pseudoallergy is very similar in its symptoms to the type 1 immediate hypersensitivity. However, in contrast to the type 1 hypersensitivity, and by the way, it's very I'm not a doctor, but I know from speaking to doctors, it's very difficult sometimes without additional tests to distinguish between the IgE-mediated hypersensitivity and the pseudoallergy. So in contrast to the type 1, which is mediated by the antigen-specific immunoglobulins E, pseudoallergy is mediated by the complement activation. And hence, there is a second terminology for the pseudoallergy, CARPA reaction. CARPA stands for the complement activation-related pseudoallergy. So what I would like you to remember from this slide is that nanoparticles can be engineered to inhibit allergy. There is a lot of research currently going on to develop tolerogenic nanomaterials and also to develop nanomaterials carrying steroids and other drugs that are used for reduction of allergic reactions. I do not have time to show you this example. So we focus this lecture on toxicity, so I will talk about toxicities. Some nanoparticles can exaggerate allergy to traditional allergens. For example, carbon black particles that dominate urban pollutions, in the air, exaggerate asthma and allergy to environmental contaminants. Then it comes to the nanomaterials used for drug delivery. Pseudoallergy is one of the most common and best studied reactions, allergic reactions. There are some rare examples of the type 4 delayed type hypersensitivity to nanomaterials and specifically to dendrimers, and I will show you 2 examples in the next slides. So CARPA, or pseudoallergy, is very common with pegylated liposomes. What you see on this slide is a data from the clinical study showing the level of the complement terminal complex in the blood of patients 10 minutes after Doxil administration. Some patients did not respond, but you see that in some patients, the level of the terminal complement complex is very high. And so these patients cannot fully benefit from the anti-cancer efficacy of the Doxil formulation. Since we speak about the complement activation-related pseudoallergy, I would like to step back and just speak a little bit about the complement to show you that dual role, of the dual interaction between the toxicity and beneficial effect. Complement is a group of about 30 proteins present in our plasma. And the main function of the complement, and at least the first historically discovered function of the complement, is non-specific clearance of pathogens. However, undesirable activation, as we've seen from the first slide, is responsible for the hypersensitivity reactions and anaphylaxis. However, in recent years, we understand that the function of the complement system is more complex and that the complement proteins and complement activation is needed for enhancement of the B cell responses to antigen, to promote T cell activation, to promote dendritic cell activation, and improve both humoral and cellular immune responses. What does it mean for nanotechnology area, and specifically for nanotechnology-based pharmaceuticals that are used for cancer therapy? If we try to use nanoparticles to deliver the drug systemically, complement activation should be avoided. Why? Because the benefit of that formulation would significantly decrease, and you've seen it on the previous slide. However, if we change the route of administration for this material to subcutaneous and intradermal, remember I showed you that these particles delivered through the subcutaneous intradermal route, especially if their size is small, travel through the lymphatic to the draining lymph nodes. These formulations would benefit vaccine efficacy, and this is very important to keep in mind. Now continue talking about allergic reactions to nanomaterials. This is an example of the study that described delayed type hypersensitivity to dendrimers. To my knowledge, this is the only clinical report, and I personally do not understand, because many people worldwide work on synthesis dendrimers. So what I don't understand, and I'm curious of knowing whether this specific patient in this case had some underlying condition or somehow was sensitive to dendrimers, or maybe some safety rules were not followed. But this is a case where a 22-year-old student in Japan, after synthesis of the dendrimers, 24 to 48 hours after he synthesized the dendrimers in the lab, developed a rash and was delivered to the emergency room with fever, chills, and this Nikolsky reaction. And he was treated with steroids, stayed in the hospital, then was discharged from the hospital. He came back to the lab, and 24 hours later, he returned to the emergency room with the same symptoms. And this continued every time he would come back to the lab. If he would not work in the lab and come back to his office, he would still be at risk developing this reaction, and the risk would increase if the cleaning of the lab coincided with his presence in the office. So he was not physically in the lab, he was in the office, but laboratory was clean, so something, whatever antigens inducing these reactions were in the airflow that connected the lab and the office. So the mechanism is unknown, but we need to, the reason I'm using this example is because such reactions are possible, and there is a huge need in research to understand what is causing these reactions, how to avoid these reactions, because many people are working with nanomaterials in the lab. We are using these materials for drug delivery, and we are exposed to nanomaterials in the environment due to the contamination of the water and air with industrial pollution. The cytokines are biomarkers for the immune cell activation. And I would like to share with you one of the examples from biotechnology therapeutics. And the reason I'm using this example is to say that we can leverage these lessons for development of the nanotechnology-based therapeutics. This is a study in Europe with a drug called TGN-1412. TGN-1412 is a CD28 supermonoclonal antibody that was tested in preclinical studies in non-human primates and in rodents, and it was safe. There was no cytokine storm. However, then this drug was injected into 6 human patients in Europe several years ago. It was injected into 6 volunteers with an interval of 10 minutes. And with the interval of 10 minutes, each of these volunteers ended up in intensive care unit and had to stay there for a prolonged period of time. Some of these patients had to lose their fingers and toes due to the massive necrosis that you see example on this picture due to the production of tumor necrosis, excessive production of the tumor necrosis factor alpha. And the very simple test using in vitro assay, utilizing human peripheral blood mononuclear cells shows that this drug induces high levels of TNF. So what we learned, I have to say that the entire biotechnology community was shaken by this accident. And what we learned from this is that we can supplement our safety studies, immunological safety studies in animals with in vitro tests using human blood. Here, I would like to show with you some similar example based on my experience at the Nanotechnology Characterization Lab. We studied metal oxide nanoparticles that have identical core, so they are made of identical material, identical hydrodynamic size, identical surface charge. The only difference was the ligands bound to the particle surface. And then these materials were tested in vivo in rats and in rabbits. One formulation was toxic, this is formulation 2, and formulation 1 was non-toxic. And the symptoms that animals experienced were very similar to the symptoms of the septic shock. However, endotoxin, which is causing septic shock, was not present in these formulations. Then we tested these particles in vitro using human PBMC. We reproduced this effect. The particle that was not toxic in vivo did not induce cytokines in vitro. However, particle that was toxic in vivo induced high levels of pro-inflammatory cytokines, TNF, IL-1, IL-8. Speaking of cytokines, I would like to share with you some of the story. You know, then I came for the job interview to the NCL in 2005. And the NCL director asked me, what do you know? Like, do you know about endotoxin, about cytokines? And I said, well, like, I know, I have some experience inducing them. I have experience detecting them. I know what is average immunologist is expected to know. But then I was hired and started working with nanomaterials. I was surprised. The first 2 years were full of surprises. And one of these surprises came with work with cytokines. For traditionally trained immunologists, we work with pathogens, right? And you know the terminology pathogen-associated molecular patterns, such as lipopolysaccharide, for example, or flagellin or CpG DNA. If we use these bacterial ligands or pathogen-associated patterns, and we add them to the immune cells, we induce broad spectrum of the cytokines. My surprise working with nanoparticles, one of the surprises was that some nanoparticles do not induce variety of cytokines, but have preference of inducing certain type of cytokines. And this is one of the examples. Lipid-based nanoparticles induce chemokines. Interleukin-8 is one example. If we see interleukin-8 induction, we see other chemokines, such as RANTES, such as MCP and MIP, but we do not see TNF, we do not see IL-1, which is very surprising. And the mechanism behind it is very interesting. So you see that here are the variety of lipid formulations. Those that do not induce oxidative stress do not induce chemokine. Those that induce oxidative stress induce chemokines. And this chemokine induction can be blocked by N-acetylcysteine, which is an antioxidant. The mechanism is very interesting behind it. So the oxidative stress results in stabilization of P38 MAP kinases, activation of P38 MAP kinases, which results in stabilization of mRNA for interleukin-8, which already exists in the cell. And cell is basically making more protein from that mRNA. It's a busy slide, but what you see here is the same. Here we are using Cremophor as a nanoemulsion example. This is induction of interleukin-8 in PBMC of various donors with Cremophor alone, and in the presence of N-acetylcysteine, we reduce it. Here we see the appearance of phosphorylated P38 to show that it activates this kinase, and we, in the presence of P38 inhibitor, we can suppress interleukin-8 induction. Okay. Another interesting cytokine which nanoparticles can specifically induce is interleukin-1. But this, then we talk about interleukin-1 induction, we talk about completely different types of nanomaterials. Now we are talking about fibrous materials and particles with cationic charge. This is a study from the literature that shows that LPS alone does not induce, at certain concentration, does not induce interleukin-1. However, when LPS treatment is combined with spherical titanium oxide particles, nothing happens. But then it is combined with the fibrous materials. In this case, they call them nanobelts. Short materials induce some interleukin-8, but the longer the fiber, the more pronounced the effect is. And the similar effects was described for the cationic particles, for example, for the cationic dendrimers. What is happening? Why cationic particles enhance production of the interleukin-1? The mechanism behind it is very interesting, and it is called proton sponge effect. Basically, what you see here is one figure I borrowed from the literature. Then the cationic material is endocytosed. Endosome fuses with lysosome. We form lysosome with excessive amount of protons. And cell tries to neutralize this by bringing more water into the lysosome. Lysosome swell and rupture. And the rupture of lysosome results in activation of NLRP3 inflammasome. There are multiple molecular components involved, but the end result of this is production of mature interleukin-1 that then cell can secrete. Nanoparticles are not immunogenic, meaning they do not induce antibody response. Then they are used even in the presence of the strong adjuvant. There are a lot of studies using fullerenes, using colloidal gold, using PAMAM dendrimers, then injected these materials even in the presence of strong adjuvant, such as complete fraud adjuvant, and did not describe formation of the antibodies. However, situation changes dramatically when these particles are conjugated to the protein carrier. So then the antibodies could be generated to nanomaterials. The take-home message from these studies is that nanoparticles behave like haptens. So they're not immunogenic by themselves, but they require protein carrier to stimulate complete immune response. And since we talk about immunogenicity, obviously it's a huge field and there are a lot of details. I would like you to remember this summary. So nanoparticles can be immunogenic. We can generate antibodies. However, so far there is no example of the engineered nanomaterials which is currently in the clinical use, either in clinical trials or approved for the therapeutic application, either by itself or carrying therapeutic proteins, which would induce either antiparticle or anti-protein immune response. Simply, there is no such example. And perhaps the reason for this is because most of these materials, remember, use hydrophilic coatings such as polyethylene glycol that masks the protein on the particle surface. Moreover, there are examples where a nanotechnology can be used to carry therapeutic proteins and reduce immunogenicity of therapeutic proteins. Pegylated gold TNF is one example. Liposomal streptokinase and liposomal factor VIII are two other examples. However, there are particles that are troublemakers. These particles are accidental. These particles belong to the following classes: glass fibers, cellulose fibers, tungsten, silicon oil, rubber, stainless steel, fluoropolymers. These are not engineered nanomaterials for drug delivery. These materials contaminate therapeutic protein formulation from the rubber stoppers, from the production lines used to produce these proteins. And these proteins can increase antigenicity of therapeutic proteins that created a lot of the problem in the biotechnology industry. So what I would like you to remember is that nanoparticles can be engineered to be non-immunogenic. They can reduce immunogenicity of therapeutic proteins. They may enhance immunogenicity and be used as adjuvants to cancer antigens, to pathogen-associated ligands, and this is what would benefit vaccines. There are nanoparticles that can enhance immunogenicity of therapeutic proteins, but these are accidental nanomaterials that should not be confused with therapeutic nanomaterials. And finally, so I'm running out of time, I prepared this slide for you to show how particle structural aspects influence particle effects on the immune system. I'm not going to go in details describing all of this. Please read this recent review article if you are interested in more details. Just on your handouts, this is how you can interpret this slide. You find the color box in the bottom, in the structure area, in the roots, and you match it by color with the box in the activity section. And you will understand, for example, here, if particles carry therapeutic nucleic acid as the API, common toxicity is cytokine induction, pyrogenicity, type 1 interferon production. Particle is cationic, or particle component is cationic. These particles commonly induce hemolysis, platelet aggregation, and the disseminated intravascular coagulation, and so forth. And I would like to finish this presentation with the following take-home messages, just to boil down to what we discussed today. Number one, nanotechnology can benefit cancer therapy by improving formulation of traditional drugs and by inducing or improving immunotherapy. Nanoparticles' physicochemical properties determine particle toxicity. Nanoparticles can be engineered to either specifically interact with or to avoid the immune system. Particle interactions with the components of the immune system can be desirable or undesirable, and desirable interactions can benefit therapies of many disorders, including cancers. With this, I would like to thank my colleagues at the Nanotechnology Characterization Lab, our supporting staff, collaborators, and thank you all for your attention.